Belle Hansen. I am the Senior Manager of Operations and Programs for the International Society for Computational Biology. I want to thank you all so much for joining us on this lovely Sunday afternoon. I'd like to take the opportunity to introduce Kim Jacoby-Moore. She works for NIH's National Human Genome Research Institute. She is an award-winning science educator, and we are so grateful for her involvement in the Youth Bioinformatics Symposium. The floor is yours, Kim.
Thank you so much, Belle. So happy to be here. I'm honored to moderate today's conversation with our keynote speaker, Dr. Ruth Nussinov. Dr. Nussinov is a computational structural biologist at the National Cancer Institute. She has been a trailblazer in the field, pioneering the conformational selection and population shift as an alternative to the induced fit textbook model to explain molecular mechanisms of recognition. Dr. Nussinov posited that population shift underlies allosteric regulation. She has many titles and recognitions. So I will list just a few of them. As she serves as the editor-in-chief of PLOS Computational Biology. She is an elected fellow of the American Physical Society, the American Institute for Medical and Biological Engineering. And she is also an elected fellow of the International Society of Computational Biology, who is hosting this symposium. So with that, I would like to welcome our keynote speaker and remind folks to please enter questions that you might have in the chat so that we can moderate our conversations for today. So thank you.
So thank you very much. Can you hear me? You can hear me. Okay, and now let me then start with the slides. Yeah. Thank you very much. So I would like to start by thanking, the organizers and all the ISCB personnel and for this invitation and this organization of the youth, which I understand is STEM oriented, so this is wonderful. And I shall be very happy then to present an overview of my own journey to computational biology. I had two options as to how to present, the talk, the bioinformatics talk. One was to just give an overview of bioinformatics and up to date, the status, the current status. And the other way was to do this, of course, in a more limited way, but in a personal way. And I thought that presenting it from a personal standpoint relates more to people because there is no way we can cover the entire field of bioinformatics. So I'm going to then describe my journey to computational biology and my journey in computational biology. So how I got to it and how have I progressed and moved across different aspects or areas of bioinformatics. So computational biology has challenges. Major or key among these is exploiting and interpreting the huge volume of biological data, which is obtained by emerging revolutionary methods, and importantly, making testable predictions testable by other computations, and of course, primarily by experiment. And importantly too, and sometimes overlooked, is to observe the patterns and make the predictions and the test in a cell-specific sequence structure function linkage, because the level of expression varies among cells, and what we relate to is specific tissues or specific cells. My personal journey, as I noted in the slide, faced additional challenges. So, a little bit of my personal biography. I was born and raised in Israel. At age 18, like all Israelis, I went to the army for 2 years. It's a must unless you have some issues, some problems. At 20, I went to college. I chose to study biology, not so much because I really was very interested in biology, but because I was influenced by my family. My father was a professor in the Hebrew University in agriculture, among the founders of the Faculty of Agriculture in the university. My aunt was a microbiologist. I didn't want to study agriculture, so I went to biology. Then I had some complications. During the first year, I got married. And at the end of the year, my husband was going to go to the University of Washington in Seattle to continue the PhD in theoretical physics, which he started at the Weizmann Institute, and he was going to go to an expert in this particular area in Seattle. Of course, I followed. I started in Seattle, and then shortly thereafter, it turned out that my husband was going to graduate and obtain his PhD in 2 years, which meant that I had to squeeze 2 years, 3 years into 2. The problem was that when I continued in biology in the university, I should back a little bit. The initial problem when I started studying biology in the Hebrew University in Jerusalem, I realized that actually I didn't like biology very much. And the reason was that in those days, biology was taught in a way that you had to memorize a huge amount. And vertebra, invertebra, the anatomy of all the different families, the classifications, etc., etc. Not the way it's taught these days. I was very unhappy with this. So when I continued in Seattle, I chose to continue in microbiology. The problem was I did not enjoy very much microbiology either, because at the time, microbiology, like the biology, involved a huge amount of memorization. You know, the different types of bacteria, and if you get sick and you have that temperature, so many days, which antibiotic do you take, or a combination of antibiotics, etc. So I was not happy there either. However, I had 2 classes that I really loved. One was in organic chemistry, and the other was in biochemistry, which was taught by 3 faculty members, 2 of whom had Nobel Prize. I then obtained my PhD my bachelor degree in microbiology, and then my husband went for a postdoc to Princeton. The problem again that I faced was that Princeton was not co-ed at the time, it was a men's school. So I went to the university which was the closest in New Jersey, and that was Rutgers, which turned out to be a great choice. They had them because, again, my husband was going to quickly go back and I was going to have a baby. So they had a non-thesis program for the master's. I graduated and obtained the master's in under 1 year. We went back to Tel Aviv, and then I stayed home for 8 years, having 3 children and raising them. At the end of 8 years, we went back to New Jersey. My husband went back on a sabbatical from Tel Aviv University, the physics department, and I came along and I decided I wanted very much to go back to school. I came to Rutgers. The problem was that at the time, Princeton was already co-ed, but I thought that I had better chances at Rutgers because Rutgers may remember me from my master's days. I came there. The problem was that the committee looked at me a little bit suspiciously, the committee for, accepting students. And they were saying something to the sort, how are you going to obtain your PhD? You're a woman, you have 3 kids. We don't see how you're going to make it. And then on top of this, I told them that again, I was constrained by time. My husband had 1 year of sabbatical. He could stay a second year on a leave of absence, but I had to finish the PhD very quickly in about 2 years. So that added to this, and they hesitated. Finally, they said, okay. I was very fortunate in going to Rutgers because at that time there was a new assistant professor there. His name is George Pichenik. George Pichenik did his postdoc in Cambridge, the UK, where he witnessed firsthand the birth of the DNA sequencing method, Unger and Coulson, and then, of course, Walter Gilbert and the Walter Gilbert method. So these revolutionary sequencing methods produced very long DNA and consequently RNA sequences. So George Pichenik then suggested a challenging problem, although at the time I did not realize that it was challenging, but he suggested that I develop an efficient algorithm to fold long RNA sequences. I didn't realize how challenging this was, so I very naively accepted. Being naive usually is not a very good thing to do. In this case, I was fortunate to be that naive. The project that he suggested faced 3 major obstacles. First, prediction should be accurate. At the same time, in those days, 1975, there were very few experimental RNA structures to test or improve their parameterization. So how would I know if my predictions are correct? Second, even if I predicted correctly, the function of specific RNA substructures were uncharted. And most importantly, computationally, it was a hugely difficult problem, which is why at the time there were no algorithms to do this. The thing is, we know RNA has 4 types of bases, A, U, G, C, and there are 3 possible base pairs, AU, GU, and GC. For long RNAs, the number of possible structures that RNA can fold is immense. So how can you generate all possible structures to find the optimal one? So this is a combinatorially hard problem. But as I say, I didn't really fully realize. So I went ahead and did it in collaboration with mathematicians also from MIT, the mathematics department, and I was fortunate to have this collaboration. The outcome is called in the internet and various papers. If you go to the internet and look, it's called the Nussinov dynamic RNA folding algorithm, which is now everywhere. I'm not going to go, of course, into the details. It's all published a long time ago and available. In addition, there was also the way I represented the RNA chain, which now again is called on the internet the Nussinov representation. So here, for example, if you look, you see on the left, RNA is drawn as a circle, and each dot is a nucleotide, A, U, G, or C. The algorithm was for the secondary structure of RNA, that's to say the planar structure, meaning that you have to find the optimal organization of the base pairs, which is shown here on the left, such that if you squash the structure, squash the circle, you will have what you have on the right. No intersection of base pairs. So this was the representation which allowed the algorithm to be, to be developed and implemented. These days, this algorithm and representation is everywhere on Google. And when I looked the other day, I saw that notes from Freiburg in Germany, in the Wikipedia, it's there, at MIT, classes on this algorithm, and again on MIT, Carnegie Mellon, and more. So I very quickly graduated and obtained a PhD and went back to Israel to take a postdoc position at the Weizmann Institute. To continue with the RNA folding, I didn't feel that I wanted to do this because I could have optimized the algorithm further, but I didn't know whether that's what I wanted to do all my life, to keep just further optimizing a little bit here and a little bit there, because the essence of the algorithm was already there. So I decided to move and to go from algorithms to fold RNA to bioinformatics of genomic DNA sequences, because as we said, these were coming out. Excuse me, I see here, stop sharing now and hide. Hide would put this box behind, right? Would it show? Yes, okay, very good. So from algorithms, so I moved from algorithms to fold single-stranded RNA to bioinformatics of genomic DNA sequences. Again, pioneering at the time, because we are talking about the late 1970s and early 1980s when these sequences arose. And what the work that I have done then, these pioneering bioinformatics of DNA was also described in these books that are there. Still, I was not totally happy because, okay, suppose I find signals, or patterns in the DNA sequences. What we are really interested in, and after all, I was coming from biology, is how do these patterns act to function? So that brought, in order to play a role in cell functions, these DNA sequences have to be recognized by proteins, which suggests that one has to take up the problem of molecular recognition. Again, was that this is a very computational. This is so because molecules recognize each other through their surfaces. So we need to explore the surface in terms of the 3 dimensions. Sequence analysis may not be sufficient because the molecules fold. It's not a sequence as when it's stretched as a string that's being recognized. So I decided to go into the detection of real 3-dimensional motifs. And this goes much beyond protein DNA recognition, because it also involves protein and protein drug recognition and more. Again, the problem at the time was that the algorithms which were available were all in terms of the amino acid sequence order. And consecutive residue patches are not necessarily adjacent. So here is the problem. On the left-hand side, so we suppose we are now looking at 2 protein molecules. and we are comparing them. And when we put one on top of the other, we see that they have a similar patch of surface. And that patch of surface involves similar positions in 3 dimensions, residue A in one molecule with residue A in the other molecule, residue B with residue B, C with C, E with E, and D with D. However, the problem is that in the first molecule, it goes, the motif depends on the order of the residues, which is A, B, C, D, E. Whereas on the right, the residues which are involved are A, B, E, C, A, B, E, C, D. So had the algorithm that would be used to detect the similar arrangement in space depends on the order of the amino acids on the chain, we would never find it. We needed then a space and another algorithm which is sequence order independent. I continued then my journey, and got to develop an efficient sequence order independent for structural carry zones and docking. Docking means matching the 2 structures like they come together. Can you hear me? Are you there?
Yes, we can hear you.
You can hear me. Okay. So, what to do? The problem again is very difficult. I was, however, fortunate again. I met before, a couple of years before, I met Chaim Wolfson, who was a computer scientist in Tel Aviv University. And one day I met Chaim on the campus. I was in the medical school. And one day I met Chaim Wolfson on the campus of Tel Aviv University. And I just asked him, Chaim, what are you doing these days? And Chaim told me that, imagine, he said, Chaim was in computer vision. And he said, imagine, Ruth, that you want to find an unpredefined pattern in aerial photographs. But the pattern can be partially occluded by other objects. For example, he was for many years in the army, so he was thinking of aerial photographs and objects like tanks. But the tanks can be partially occluded by trees, by other vehicles, etc. So the patterns have to be detected when they occur with what we called errors. Errors means mismatches. When I heard that, I literally jumped because it was very clear that even though Chaim was coming from a very different area from computer vision, the problems were actually quite similar, even though in biology, as always, they're more complex. We were dealing not with 2D, but with 3D. And our mismatch, our so-called errors involved amino acid, substitutions or deletions or insertions, in short, various mutations or changes. Still, the basic algorithm could be taken and adapted to our problem. And this is exactly what the many students that we had in Tel Aviv University over the years have done. Later, I mentioned to Chaim that actually the molecules are flexible. They have hinges. And one part of the molecule can move with respect to the other part. What do we need to also handle these molecules? Chaim then showed me in his office that before, what he has handled when he was in IBM was what he called in various, objects with hinges in cluttered scenes when they are sort of put one on top of the others. And these objects had hinges like scissors, garlic squashers, pliers, etc. But you still want to be able to detect that you have pliers here, pliers there, scissors here, scissors there. Well, again, in proteins, it's much more complicated, but still the robotics approach that he took with those objects was not unsimilar. So we developed the algorithms and adapted the applications to these diverse areas. Then I wanted very much, after all, I'm a biologist, I wanted to apply it and to find what is the preferred way for proteins to interact. This is a very difficult problem because what you need to do is if you have, say, two protein structures and you look at the surfaces, there are many, many ways that you need to try to match one with the other because you have, in essence, to try and match every patch of surface of one molecule with every patch of surface of another molecule in 6 dimensions, 3 rotations and 3 translations. But we would like to know, our ultimate goal is to predict the preferred mode of protein associations. And we know that similar protein structures can associate in different ways. It's enough to have few mutations which are on the surface to change the patch of surface. And at the same time, different protein structures can associate in similar ways simply because these particular ways are very favorable energetically and evolution has conserved them. So I continued this time with my wonderful colleagues in Koç University in Istanbul, Özlem Keskin from the chemical engineering, and Attila Gursoy from computer engineering in Koç University. And their students, again, very long-term collaboration, still ongoing. Question is then how to predict the preferred way for proteins to interact. There have been 2 major strategies to do this. One is docking, which means that you start from basic principles. All you have is the structures of the 2 proteins, and you may have some additional biochemical data related to their preferred interactions, to their, not preferred, but indicate some knowledge of their interactions. And the second is knowledge-based, based on protein-protein interaction data. Protein-protein interaction databases form the basis for mapping pathways and the basis for a more complete picture of their. And within these, protein-protein interface data set is particularly useful. This is because the number of motifs is limited in nature. Consequently, a protein-protein interface data set is useful for predicting protein for the construction of interaction networks and for large-scale proteome docking, particularly when combined with refinement and energy scoring, which is what we have been doing. So proteins interact through their surface. Here is an example of a primer, a complex of 3 molecules, and you see their surfaces. I don't know what happened here. Anyway, you see my, I don't know why. Okay, so you see this is one surface right here. And this is the second surface right here. So an interface, according to our definition, includes residues in contact, one, say, from here, this protein, one from here, and residues which are nearby their interaction. And we included the nearby simply so that the prediction is more robust. So, as we said before, interfaces are actually surfaces and they do not consist of continuous fragments. So there is no sequential order. Look, for example, at this interface between this protein and this protein. If you look at the actual residues that form this interaction, we see here the number 33 to 41, 60, 61, 63, 67, 153, then 72, 83, and so long and so forth. So we can see any algorithm, any computational method which goes by the sequence of the residues on the chain would never find this particular interaction. But our algorithm that I developed together with Chaim Wolfson, the computer vision-based algorithm, which is called geometric hashing, could do this. How did we do it with Özlem and Attila? We start with the entire PDB, that's the experimental databases. Now there are also models there, mostly crystallographic. So here we see monomers, we see dimers, we see trimers. Next, we then highlighted, we marked all two-chain interfaces here in. Then we extract the two-chain interfaces here. We carry out an all against all structural comparisons using the geometric hashing, which then leads to the clusters which contain similar interfaces. And then here are, when we looked at the clusters that we obtained, we realized that we have three. The first, type 1, were not surprising at all. They contained complexes, protein-protein interaction complexes, where the structure of proteins are similar. Here, this one, similar to that one, this one to that one, and similar function. So it's expected that their interfaces would be similar. Type 2 was much more interesting because here we had similar interfaces, the structures of their interactions, the surfaces. But if you look at the global structures of the proteins, they are different. This one is different than this one, the entire protein, and this one and this one. So this is the type that we were particularly interested in, although there is also type 3, which is also very interesting, where the interface clusters had only one similar side here, the one in orange. So in type 2, we had similar interfaces and different functions. So type 2 is similar to protein structures, to monomer structures, where evolution has utilized good, favorable folds for many different functions. What it meant is that of all the combinatorial possible ways for different monomer structures to associate, they still prefer to interact in similar ways to yield preferred interface architectures. And these preferred architectures are those that are energetically favorable. So here we can see we have clusters with similar interfaces. The boxed one, this is similar to that. But if you look at the structure of the global structure of the are very different. This protein, the structure very different than this one, and this one very different than that. And at the same time, also the functions are different. Here is another example. Again, this one is similar to that one, the interface. The global structures of the chain are different. This is different than that. This is different than that. And the functions are different. And the last example here, for example, this is similar to that. The global structure of this different than that. This is different to that. And the functions are different. One is in this case a coenzyme domain for a scaffolding protein. The other is the red fluorescent. So type 2 with similar interfaces and different global structures allowed prediction of protein interactions across signaling pathways and the proteome, which we have done. So the prediction, we start with the representatives as templates. These are our clusters that we obtained. These are our targets, all the monomers in the PDB. So suppose we find that the surface on A is similar to the surface on F, and the surface on B is similar to the surface on E, on G, and on D. So then we can predict that A can interact with E, with G, and with D, and F can interact with B, with D, with E, and with G. Then we filter them, the solutions, refine them, rescore them, and re-rank them, and check against experiments. I still continued my, my journey in computing. This time I moved to mechanisms and pharmacology. So we looked at the regulatory mechanisms and pharmacology, focusing on the regulatory mechanisms and pharmacology of RAS signaling. Why did I choose RAS signaling? Very simply, there is a very high incidence of mutations in RAS proteins and in their major pathways in cancer, and drugs are still limited. And I should say that this particular problem at the NCI, and not just the NCI, across the scientific, our relevant scientific disciplines. And it's not just biology, it's genetics. Here we are doing also, it's biophysics, it's experimental, it's computational, it's many physicists have taken it up, and more. So here is some statistics of RAS and the KRAS mutations. RAS has several isoforms, that's to say variants. The major ones are HRAS, NRAS, and KRAS. Looking at these, we see the incidence of more, of course, than. And here, just the KRAS mutations, the distributions of their major mutation at glycine 12, glycine 13, and glutamine 61. So here is the statistics of RAS isoform mutations in human cancer, which just shows us how much we still do not understand. KRAS, in particular within the KRAS, its splice variant, KRAS 4B, has a 95% incidence in pancreatic cancer. Why particularly KRAS? We still do not fully understand. In colorectal cancer, KRAS, again, mostly KRAS 4B, has a 45% incidence in cancers. In lung adenocarcinomas, KRAS again, at 35%. Again, we still do not understand why. AML, 30%, NRAS isoform. Melanoma, 15% NRAS. Again, we do not understand why. And in bladder cancer, and also cancer of the head and neck, HRAS is the most prevalent, although the percentage is much lower. I and my group were very interested in figuring out why these particular mutations. So, in going into into the structures, the interactions, the signaling, etc., of these particular of RAS protein. In particularly, we have been focusing on KRAS4B for obvious reasons and its mutations. So there are still major knowledge gaps in RAS. There are not all structures of mutant KRAS proteins with their effectors, that's to say, with the proteins with which they interact and initiate the signaling downstreams, and regulators, those that regulate them, are available. We don't know which effectors are important in established cancers and their cell-specific signaling. The signaling complexes and the regulation is still not fully characterized. We don't know why different alleles have different clinical outcomes, and we don't know which cancers are RAS. Finally, only recently, the first successes in targeting KRAS cancers have been achieved primarily through, not primarily, yeah, I guess primarily through again, showing how much we still do not understand. Here are KRAS mutations in 3 diseases. The statistics with here the G12 mutations, G12 where the glycine is substituted. Am I over time?
No, I just wanted to let you know we have about 5 minutes left, and then we have a couple questions queued up for you already.
Okay, please let me know when I should stop. Okay, so I'll just say, I'll try to go a little faster, that there was the question, why different mutations have different incidences in different cancers. See, for example, in the pancreas, G12D. So we don't understand why. In lung, G12C. In colorectal cancer, G12D, we do not understand why, and this is what we have been focusing. What we have, and I'll stop with this, what we have now is the knowledge of, we have diagrams, which are, so here, for example, we have RAS, proteins that activate it with arrows, proteins that inhibit it, but with bars. So we have these diagrams. However, these simplified diagrams, which are highly informative and provide an extremely useful overall picture, they still are unable to capture the root cause of pathological alterations, which is what we were interested in. So I'm going to skip the various questions, or observations, why, and what needs to be activated. And I think I'm going to this is regarding drug discovery, what we have done, the activation, nanoclustering, and I'll only end by saying that computational biology journey still continues and continues. Currently, in addition to RAS and its pathway, which is a major project in my lab, is looking at the human pathogen protein-protein interaction, trying to figure out how pathogens can take control of the host cell. Hijack, and again, we use the same protein-protein interaction algorithm with some variant to fit into this. And here is the basics of changing of the network, which the when we used interface architectures, again, to date, all other available approaches are sequence-based. And here is one example, which I skip over, and end by acknowledgements. Here, my colleagues at the NCI, Hyun Bam Zheng, Zhen, Tsai, Emine, Shi, and Young, who have by now left. At Koch University, Oslem, Attila, and their students. In Shanghai, Xiaoyong, Shuai, the group. University of Illinois, this is Russ, Im Gapunenko, and his students. At the NCI also, additional colleagues. At Northeastern, the crystallographer, Carlo Matus, University of Maryland, David Fushman, and our joint student in Belgium, Anna, and David Sachs at the Clinical Center of the NIH. Here in Tel Aviv University, primarily Chaim Wolfson, and I listed some of our students, many students that we had, 20 students or so. More recently, I've started a wonderful collaboration with Faixiong Cleveland Clinic, and with him it continues now also. And with that, I shall close, and I'll be happy to answer any questions.
Thank you for sharing your personal journey and your career exploration story with us, Ruth. It was really quite fascinating. We already have a few questions in the Q&A, so I'll just go in order. From one student in New Jersey, they wanted to know, do you think all human proteins have been identified, or are there still more left to discover?
This is a very important and interesting question. I think most have been, but most likely still some are not. That's my feeling. Proteins are still but identified, yes. But I think it is the more acute problem is also, it's not just the discovery of the protein, which are generally variants. We know, I mean, there are expected to be about 30,000, but it's the interactions of the proteins, because if we don't have the interactions of the proteins, we cannot figure out how they function. So it's the function, all these areas, as I hope came through, it's very important. Function, cell type specific function.
Well, that leads nicely into our next question. Why do RAS protein mutations frequently fuel cancer progression?
Excuse me, I did not get this question. Why what?
RAS protein mutation frequently fuels cancer progression.
Cancer progression, we do not look at cancer progression. The cancer is such a hugely complex problem. We have been focusing on cancer initiation, not progression. Progression, then you have to come to, you come to, it undergoes so many, it's the resistance, and then in multiple tissues, and all the changes that are that the cells undergo, the additional mutations, either in the same protein, the original proteins which we have been looking, additional mutations. It's not one. I did not go into this at all. Generally speaking, there is the single mutation initiation. There is single mutation, but this is not the whole story. Mutations, cancer does not arise from a single mutation. There have to be several mutations, which is why older people have a higher chance of getting cancer because they accumulate mutations over the years. So there can be 2 mutations, 3 mutations, or more. And the mutations can be in the same protein or different proteins in the same pathway or different proteins in other pathways. And then the key question, which we are very interested in, is the effect, and can we predict which in drug resistance, which protein and which pathway will be affected next?
Thank you so much. Earlier in your presentation, you talked about long genomic sequences in the 1970s and 80s. How do they compare to long read sequences for technologies like nanopore today?
Excuse me, how do they compare to what?
Long read sequences today, like through using nanopore.
Nanopores? That's a very different question, a very different aspect, very different, yeah. We did look, at some nano questions, but we have not been following up on this. There are so many problems in biology, and nano is extremely important problem, but I would say it's not the same, it's different. And usually it comes from aggregation. It's nanostructures are self-assembly usually, so it's just different. Here we don't, of course, self-assembly, that's complexes, but nano, you have many building blocks. We looked at it a little bit, in the context of Alzheimer, which is self-assembly.
I think it's so admirable what you have been able to accomplish in the span of your career. Do you think that your PhD advisor or even your postdoctoral advisor acknowledged the challenges that you overcame both computationally for your research projects, in addition socially, in probably being one of the few women in that space?
So first of all, let me yeah, okay. So let me say that here now I can see you. This is better. So early on I did not go into this, but when I joined Tel Aviv University, the medical school, also I talked about my early problems, not problems, but challenges. When I joined the medical school in Tel Aviv University, it's hard to imagine that these days such comments or questions would be made. But at the time, the dean called me. He said that I'm a mathematician. I said not at all, and I would be happy to have been one, but I'm not. So later on, he came with a problem he wanted me to help him figure out. Times change. I think these days, computational biology, bioinformatics is broadly recognized as an essential field. Just think of the huge amount of data, what we call these days big data, right? So many people are involved in generating it. But it's not going to be useful without analysis of this data. And to analyze this data, to compile, to analyze, to sort, etc., you need efficient algorithms. Right? So there is no question. I know the NCI, for example, started a laboratory for data science. We know that the bioinformatics, and not just, I try to emphasize, we call it bioinformatics, but it's really computational science broadly, because I also touched on computational biophysics, which is involves structural biology, computational structural biology. So I think this particular area is vastly important, not to mention drug discovery. I did not go into it at all. I'm now involved, in drug discovery. There is an initiative in the NCI as well. So it's all very, and we know, for example, machine learning, AI, a whole new setup of challenges which can help in faster and more efficient outcomes. So yes, all of this is very important, and I think computational biologists, bioinformaticians are going to be in higher and higher demand. But the key is not, which I tried to emphasize throughout my talk, is not to start with one problem and necessarily stick with it throughout your career.